Welcome back, my dear students. Did you know that the secret of life lies inside tiny structures called chromosomes? They carry all the information that makes you who are you. Today, we will explore the structure of chromosomes, understand the amazing journey of the cell cycle, and see how cells multiply through cell division. These processes are the key to growth, repair, and even heredity. So let's dive in and unlock the mystery of life's blueprint together. Our syllabus includes cell cycle and cell division, structure of chromosomes. We should study mitosis and its stages, basic understanding of meiosis as a reduction division. Stages are not required here. Brief idea of homologous chromosomes and crossing over leading to variations. Significance and major differences between mitotic and meiotic division. Basic structure of chromosome with understanding of terms such as chromatin, chromatid, gene structure of DNA, and centromere. Importance of cell division. Cell division is a fundamental characteristic of life that ensures perpetuation across generations. It occurs in both simple organisms like amoeba and complex ones like humans, elephants, and trees. During mitosis, duplicated chromosomes are evenly distributed to daughter cells, and it ensures cells in the body have the same type and number of chromosomes. It maintains normal functioning of cells and survival of the organism. What are chromosomes? The nucleus contains DNA organized into chromosomes. Each chromosome has one long DNA molecule associated with proteins, and this DNA protein complex is called chromatin. In non-dividing cells, chromatin appears as long, thin, slightly stained fibers. As cells prepare to divide, chromatin condenses into distinct thick chromosomes, and chromosomes can be stained and observed, hence the name. Chroma means color and soma means body. So chromosomes are highly coiled ribbon-like structures formed by condensation of chromatin fibers during cell division. Discovery of chromosomes, they were basically fundamentally first studied in 1882 by Walter Fleming, who was a German scientist. This is Walter Fleming. He is Walter Fleming. They observed in salamander larval cells, used a strong microscope and dyes, and he showed thread-like structures dividing lengthwise. He introduced the term mitosis, Greek mitosis, thread-like structure. This is the thread-like structure, and he named the process as mitosis. The above image, which is a sequence of three diagrams showing how chromatin fibers condense into chromosomes during cell division or mitotic prophage. The first figure shows a nucleus with loose chromatin fibers, the second figure shows more condensed fibers, and the third shows highly condensed chromosomes. The diagram this illustrates the progressive packaging of genetic material. The chromatin is made up of two substances, which is DNA about 40% and histones, a type of protein which is nearly about 60%. Now we should study molecular structure of DNA. The DNA double helix was first studied by Rosalind Franklin in 1953 using X-ray diffraction. Watson and Crick worked out the full structure in the same year and proposed the double-stranded helical model. They were awarded the Nobel Prize in 1962. So DNA is a macromolecule and made up of two complementary strands wound around each other in a double helix. So what are the DNA composition? Each DNA strand is made up of repeating nucleotides consisting of three components: phosphate group, sugar, and nitrogenous base. Four nitrogen bases are listed: adenine or A, guanine G, cytosine C, and thymine T. A here pairs with T, there is two hydrogen bonds, and G pairs with C, there are three hydrogen. DNA at a glance. Basically, there are two kinds of nucleic acids, DNA and RNA. The components of chromatin are DNA and histones. So the components of a nucleotide, pentose sugar, phosphate group, and nitrogenous bases. Nitrogenous bases are of two types, purines and pyrimidines. Purines are either adenine or guanine, and pyrimidines are either thymine, cytosine, and uracil. So the nitrogen bases in DNA are adenine, guanine, thymine, and cytosine, and nitrogen bases in RNA are adenine, guanine, cytosine, and uracil. Basic structural and functional unit of a nucleic acid or DNA or RNA is the nucleotide, and the chromosome is the nucleosome. So chromosomes were discovered by Walter Fleming. The shape of DNA molecule was discovered by Rosalind Franklin, and the double helical model of DNA was discovered by Watson and Crick. So the histone proteins, they are the proteins that help coil and package DNA into structural units called nucleosomes. Each nucleosome contains 8 histone proteins around which DNA is wound like thread around a spool. The nucleosome is compared to a bead on string structure. A single human chromosome may contain about a million nucleosomes. This is the image showing the concept of coiling and supercoiling. This is coiling, and this is further coiling is called as supercoiling, with a telephone cord like structure representing how chromatin fibers fold to form chromosomes. A step-by-step schematic of DNA condensation. DNA strand, it is wrapped around histone proteins. It forms nucleosome. It's a beads on string model. Nucleosomes coil. It forms chromatin fiber. Chromatin fiber supercoils. It forms the chromosome. At the cellular level, these condensed fibers are organized into visible chromatid inside the nucleus. Structure of chromosome. This structure, the image shows that the explains in condensed form visible during cell division, and each chromosome consists of two sister chromatids which are joined at a point called as centromere. This is two chromatids and they are joined at the point called centromere. The centromere is described as a small constricted region, and the functions of the centromere, it holds the chromatids together. It serves as the attachment point for spindle fibers during cell division. As spindle fibers contract, chromatids separate at the centromere and move to opposite poles of the dividing cell. Here, chromosomes decondense back into the fine chromatin fibers inside the nucleus. The number of chromatin fibers equals the number of chromosomes. So one vertical half of duplicated chromosome is a chromatid, and two identical chromatids joined by a centromere and are called sister chromatids, which separate during anaphase stage. Functions of centromere, they attaches chromosomes to spindle fiber during cell division. They help detach sister chromatids during anaphase. What are genes? Genes are defined as specific sequences of nucleotides on a chromosome that encode proteins which express particular features of the body. Genes here are described as units of heredity. They are passed from parents to offspring, determining specific traits. DNA fingerprinting, this is the image showing the fingerprinting of DNA. 99% of DNA is non-coding but highly variable between individuals. This variability is used in DNA fingerprinting, which has applications in criminal cases, paternity or maternity testing, and identifying biological evidence from blood, semen, or hair. Need for new cells. What are the need for cell division? For growth, a new organism starts as a single zygote. This is the fertilized egg. The zygote then divides repeatedly to form a cluster of cells which specialized into tissues and organs. For replacement, it explains wear and tear. About 2 million red blood cells die every second. These are replaced by new cells formed by division of parent cells in bone marrow for RBCs. In plants, old leaves fall off and are replaced by new ones. For repair, tissue damage for injury, that is cuts or bone fractures, is replaced by cell division, where new cells fill the gap. For reproduction, cell division is also vital for reproduction. In simpler organisms like amoeba or bacteria, cells fundamentally divide by mitosis to produce identical daughter cells. And in higher organisms like plants and animals, reproduction involves specialized cells, sperms and eggs produced through meiosis. These gametes carry only half the chromosome number of parent cells, that is haploid or N, and the fusion of gametes restores the diploid 2N number in the offspring. Number of chromosomes in humans. Body cells, somatic cells 46, that means 23 pairs in both men and women. Sex cells or gametes 23, that is single set in both sperms and eggs. Fertilized egg or zygote 23 from sperm and 23 from egg, that is 46 or 23 pairs. So the new baby, and it also has 46 chromosomes, that is 23 pairs diploid. So cell cycle, it divide, grow and redivide. The cell cycle ensures that a cell grows, synthesizes materials and duplicates its DNA and divides into two daughter cells in an orderly manner. So it can be defined as a series of events leading to DNA duplication and cell division. It consists of two main phases. Interphase means non-dividing phase where growth and preparation occurs, and M phase, that is mitosis or dividing phase, where the cell actually divides. Interphase. They grow to the same size as the mother cell with active nuclei but less cytoplasm. Interphase is not a resting stage, so the cells are metabolically active and they synthesize materials needed for division. Three phases of interphase are: first, growth phase or G1, RNA and proteins are not synthesized, volume of cytoplasm increases, mitochondria in animals and chloroplast divide, and at the end of G1, cells either enter the resting phase or proceed to S phase. Synthesis phase, DNA is duplicated and chromosomes create identical copies of themselves or sister chromatids. Second, growth phase or G2, here more RNA and proteins are synthesized, prepare the cell for actual division, and the cell is not ready to enter mitosis. Cell cycle regulation. The cell cycle can stop permanently in some places, temporarily in others, and resume only when needed. Regulatory mechanisms control when cells divide or stop. The examples of cell lifespan: nerve cells, that is brain or cerebral cortex, and eye lens, and most muscle cells once formed in the embryo, they are not replaced after dying. And the skin cells, they are replaced in every 2 weeks. Red blood cells, they live for 120 days before being replaced. Bone marrow, they are replaced every 10 years. Epithelial cells or lining gut replaced every 5 days. Liver cells, they are replaced every 1 to 2 years. Other body cells on average 15 years of lifespan. Cell production and death. In children, more new cells are produced than those dying. Their growth occurs. In adults, production equals death and the cell number remains constant. Fewer new cells are produced and cell death surpasses production. Cell division. Two types are introduced. Mitosis produces diploid cells for growth and development, and meiosis produces haploid cells, that is gametes like sperm and egg. Mitosis, the word mitos meaning thread, we have already mentioned it and referred to the chromatin threads. Definition: a type of cell division where one parent cell divides into two identical daughter cells. A key point: chromosome number remains the same at each division. Steps of mitosis: it can be divided into two processes: karyokinesis or nuclear division, and cytokinesis or cytoplasmic division. Karyokinesis has four continuous phases like prophase, metaphase, anaphase and telophase. This is the four successive process. Interphase, it is also called as resting phase. So they can be discarded as the described as the stage where the cell is not visibly dividing. No visible change in chromosomes, but active DNA synthesis is ongoing in preparation for division. So what is prophase? During prophase, chromosome changes. Chromosomes become short, thick, and visible, and each chromosome has already duplicated, forming two sister chromatids attached at centromere. Centrosome activity: in animal cells, the centrosome splits into two and centrioles move apart together opposite poles of the cell. Each centriole is surrounded by radiating microtubules called asters, that is the star-like structures, and plant cells do not have centrioles, so asters are absent. Spindle fiber formation: fibers appear between the two centrioles forming the spindle apparatus. Nuclear changes: the nuclear membrane and nucleolus gradually disappear. movement of chromosomes, the duplicated chromosomes start migrating towards the equatorial plane or middle region of the cell. Now we'll study the metaphase. Uh, it is the next stage after prophage. Key points: duplicated chromosomes align on the equatorial plane. This is the middle of the cell, and each chromosome attaches to spindle fibers by its centromere. So this is the diagram showing the interphase stage or resting stage and prophage in the figure A and figure B. Figure A indicates the animal cell and figure B indicates the plant cell. This is also a diagram showing the metaphage, anaphage and telophage and cytokinesis in both animal cell and plant cell. Anaphage, ana means up or back. So here in this process the centromere attaching the two chromatids divide. The two sister chromatids of each chromosome then separate and are drawn apart towards opposite poles. The drawing action is performed by the contraction of spindle fibers. Telophage, telo means end. Each chromatid or the daughter chromosome's length then becomes thinner and turns into a network of chromatin threads. Nuclear membrane reappears. Nucleolus reappears in each daughter nucleus. So in this process all the contents of the cytoplasm including the mitochondria and also the chloroplast in plant cells are randomly distributed in daughter cells during karyokinesis. Cytokinesis, division of cytoplasm. At the end of telophage, a furrow appears in the cell membrane in the middle which deepens and finally splits the cytoplasm into two, thus producing two new cells. Karyokinesis and cytokinesis, all the nuclear changes that occur during cell division are collectively called karyokinesis. Karyo means nucleus, and karyokinesis is followed by the division of cytoplasm or called as cytokinesis. Mitosis in animal cells and mitosis in plant cells. In animal cells, asters are formed. In plant cells, asters are not formed. In animal cells, cytokinesis occurs by furrowing of cytoplasm. The furrow starts from the cell membrane and progresses inward toward the center of the cell. In case of plant cells, cytokinesis occurs by cell plate formation. The cell plate forms in the center of the cell and extends outward to the cell membrane. Occurs in most tissues throughout the body for growth and replacement. Mitosis in plant cells, it occurs mainly in meristematic tissue, at growing tips for lengthening and at sites for increase in girth. Spindle fibers are formed with the help of asters and centrioles. Spindle fibers are formed with the help of microtubules. So what are the significance of mitosis? Growth or increase in the body size due to formation of new cells in the tissue, repair of damaged and wounded tissues by the renewal of the lost cells, replacement of the old and dead cells such as the replacement of the blood cells and the epidermal cells of the skin. Asexual reproduction in which the unicellular organisms such as amoeba or the yeast cells divide into two. This process of mitosis maintains some chromosome number in the daughter cell. So here the diagram shows the sectional microscopic view of a plant cell undergoing active mitotic cell division. The various stages are displayed here. Interphase stage, prophage, metaphage. The 4 is the anaphase stage, and 5 is the telophase stage, and 6 is the young cells formed after cell division, after cytokinesis. So mitochondria and chloroplasts in cell division. During cell division, both mitochondria and chloroplast have their own DNA which carries specific genes. So they also contain their own ribosomes, allowing them to produce some of their own proteins. These organelles divide independently by a process similar to binary fission, that is splitting into two. During mitosis, they are partitioned between the two daughter cells so that each cell receives the necessary organelles. However, mitochondrial division is also influenced by genes present in the nucleus and coordinated with the cytoplasmic ribosomes. Introduction to meiosis, reduction, division producing gametes. The term meiosis is derived from the Greek word meion, means to lesion or referring to the reduction of chromosome number. So here the meiosis can be defined as the type of cell division that produces sex cells or gametes. In human, it occurs in testes to produce sperms and ovaries to produce ova. In flowering plants, it occurs in the anther, producing pollen grains, and ovules, producing egg cells. Overview of meiosis. Meiosis states that meiosis is completed in two divisions. Meiosis 1, it is the reduction division, chromosome number halved, and meiosis 2 is the mitotic division, it resembles the mitosis. It also notes that G2 phase is absent in meiosis, and the prophase of meiosis 1 is the most similar to prophase of the mitotic cell cycle. So what is the significance of meiosis? Reduction of chromosome number. Gametes or sex cells have the half the number of chromosomes. During fertilization, the normal diploid chromosomes is restored. Mixing up with genes, genetic variation happened in two ways. Maternal and paternal chromosomes mix during the first division as homologous pair separate. Crossing over occurs when chromatids from maternal and paternal chromosomes exchange their segments. Crossing over. Exchange of genetic material between non-sister chromatids of homologous chromosomes during meiosis. Chiasma. The egg-shaped structure formed at the point of attachment between two non-sister chromatids where crossing over occurs. Importance: it creates new gene combinations, it leads to genetic recombination in offspring. It explains why siblings from the same parents differ in many traits despite their genetic similarity. So this is the difference showing the process of mitosis and meiosis. This is the summary of the chapter. So my dear students, chromosomes are like the library of life, storing all the instructions that make every living thing unique. The cell cycle is the cell's way of preparing and working in perfect order, and cell division is the magic that helps us grow, heal, and even pass traits to the next generation. From mitosis to meiosis, we discovered how cells keep life growing strong. Thanks for joining me on this journey inside the cell. If you enjoyed learning today, don't forget to like, share, and subscribe for more simple and fun biology lessons. Keep your curiosity alive and keep growing like a cell. See you in the next class. Stay happy, healthy, and blessed. Thank you.